Now it's my distinct pleasure and honor to introduce Dr. George Daley of Harvard Medical School to the Museum of Science. Dr. Daley is an associate professor of pediatrics at Children's Hospital Boston and biological chemistry and molecular pharmacology at Harvard Medical School. He is also associate director of the stem cell program at Boston Children's Hospital and a member of the executive committee of the Harvard Stem Cell Institute. This is a new institute. Dr. Daley's research is aimed at translating insights into stem cell biology into cellular therapies for degenerative, malignant, and genetic diseases. Along with Dr. Rudolf Janisch, his laboratory reported the first successful application of therapeutic cloning of embryonic stem cells to treat genetic diseases in a mouse model of immune deficiency, and also the first creation of functional sperm cells from embryonic stem cells, work that was cited in Science magazine as a top 10 breakthrough for the year of 2003. We are truly honored to have such an accomplished scientist with us here tonight to present one of the hottest topics in science today. Please join me in thanking Dr. Daley for the gift of his time and talent.
Thank you very much, Cheryl. This is a special evening. It's very, very distinctive in many, many ways. It's a delight to be here at the Museum of Science. The Museum of Science is one of the great institutions of Boston. It's one of the great institutions of all of science, and its mission is really to be applauded. I'm really pleased to be part of that. I'm also quite honored that several members of my laboratory are here tonight, sitting in the front row, Claudia, Kiwi, Willie, Holm, Azam, and they will be mingling afterwards and helping to answer questions in a one-on-one basis. I also have to say that it's the first lecture I've ever given with my mom in attendance. My mom, who is really so responsible for much of my intellectual curiosity, which is what I hold so dear to me and has really launched me into my career as a scientist. And it's wonderful to be able to thank her publicly. And it's also the first time I've ever given a lecture where they're serving alcohol. So I hope you all drank deeply beforehand. Perhaps what I'm about to say will make more sense. The topic of stem cell research has gotten an enormous amount of attention. And it's precisely because it is so important to us as a society, as we move forward, to think about science in a social context and to weigh the enormous potential against the many, many pluralistic ideas that we hold in this society. And it's that debate which I think is what makes our country so great. And I hope today, this evening, I'll be able to describe to you why the science is so important and why I believe firmly that the right answer is to continue to move forward in this exciting new realm of biomedicine. The folks who are seated up front are a small part of my laboratory, many of whom are pictured here. These are young people who are devoting their careers to the study of science, to the probing of the frontiers of knowledge, to the ultimate development of the next generation therapies for diseases like cancer and immune deficiency and sickle cell anemia. And I'm pleased that you'll have a chance to get to meet at least some of them this evening. I owe an enormous debt to them. They're a terrific, terrific bunch. They make my job one of the greatest jobs on earth. What I'll talk about primarily this evening are embryonic stem cells. I will talk about adult stem cells, but primarily focus on these fascinating cells that are immortal and can be grown in a petri dish indefinitely, and which offer so much promise, both for addressing fundamental questions of science and perhaps ushering in what I believe will be a new era of medicine, an era of medicine where we will add to our armamentarium of drugs the ability to deliver cells as medicines. And as you appreciate, our society is getting older. And with an aging society come a whole new realm of diseases of organ and tissue failure. And diseases like heart failure, kidney failure, Alzheimer's, Parkinson's, and the like are not easily treated by drugs. Once tissue function is lost, the best way to repair those diseases is to replace the diseased organ. Now, as you can appreciate, there's enormous shortages of donor organs for the treatment of kidney and heart failure and liver failure and the like. Many organs simply cannot be replaced. And one great vista for the future is to imagine replacing cells. And one of the most important potential sources of those cells are embryonic stem cells. Now, most of the hype surrounding embryonic stem cells, most of the promise, if you will, has to do with the potential to learn how to coax these cells, which, as growing in the Petri dish, are unspecialized, but can be coaxed to become specific cell types that might be delivered into a diseased patient to repair the bone marrow, parts of the central nervous system, a failing heart, or perhaps to replace the insulin-producing cells in the pancreas of a diabetic. Now, this is much of the hype, but I want to make the point, and I'll return to this point again and again, that even if embryonic stem cells are never successfully applied in clinical therapies, actually being transplanted into patients, that they are enormously valuable research tools. And just as tools, they are valuable and should be studied. We know from 20 years of experience studying embryonic stem cells from the mouse that these can be used to give us a window on early aspects of development, and in the case of human embryonic stem cells, teach us about human development, and are being increasingly accepted by the pharmaceutical industry as tools that allow us to define new drug targets and might potentially even be used for the development of new drugs and toxicity testing. But of course, the promise I think everyone hopes for is in tissue delivery for cell therapy. The question I'm often asked is, how long is it going to take to realize this promise? Well, if you read the literature, you realize that stem cell therapies are being delivered today. In fact, the stars among us are already exploiting this bizarre new therapy to beat aging. Now, I think this tells us how challenging this is going to be, because I leave it to you to determine whether or not this is a successful application of stem cell therapies. I think we're hoping to do better. Let me start with a definition of a stem cell. When a scientist talks about a stem cell, they have a very simple set of criteria. They call it a single cell that can divide and become copies of itself, the concept of self-renewal. One of those daughter cells has to also be able to undergo continued specialization to develop into a set of distinct differentiated cells. That's all one has to be called a stem cell. You have to be able to self-renew and differentiate. But all stem cells are not alike. There is indeed a developmental hierarchy of stem cells. The stem cell that we know most about exists in the bone marrow. It is the stem cell for the blood. This is a single rare cell, 1 in 10,000, 1 in 100,000 cells in your bone marrow, that alone is responsible for the production of all the red blood cells, white blood cells, and platelets that make up your blood system. We know more about stem cell biology from studying this cell than any other, and this cell has been delivered in therapeutic bone marrow transplantation successfully to cure leukemia and genetic disease for the better part of 3 decades. The principles that allow us to define the self-renewing and multilineage hematopoietic or blood stem cell have taught us to define stem cells for other tissues. So we appreciate that there is a stem cell for the connective tissue, the gristle of the body, the bone, the muscle, the fat. There are stem cells in parts of the brain that can produce not only neurons, but the supportive cells of the central and peripheral nervous system. It is believed, or hypothesized at least, that there might indeed be stem cells for all the tissues in the body. There is no doubt, and it's been definitively proven, that there are stem cells for the skin, there are stem cells for the testis, there are stem cells for the gut. Slightly less well established are whether there are stem cells for the liver, and in fact, recently, there's been a transformation in our thinking about the pancreas, whereas previously we had believed stem cells existed. Doug Melton, one of our colleagues at Harvard, has recently published a very provocative and profound paper suggesting that at least for the islets of the pancreas, those cells that give rise to insulin, that in fact these are not replaced from stem cells. So there's an important principle here. The very rapidly growing tissues of our body, those that are affected most by chemotherapy, the blood, the skin, and the gut, clearly actively replenish from stem cells. But many other tissues of the body, the brain, the heart, much of the kidney, and perhaps other tissues, may not actively be sustained by stem cells. This is a very, very important point. Now, these are the adult stem cells, and these are to be contrasted with the stem cells of the early embryo. The master cell of us as an organism is the zygote. This is the single cell that's created when the egg and sperm come together. A new genome is formed, and within that single cell is all of the information for creating the miracle of human development, really, which is the body plan of a complete adult organism. This zygote, after just a few days, will become what's called a blastocyst, and this is the stage of very early human development where you're existing as only 50 to 200 cells, and it's this stage of embryonic development which is creating much of the controversy. Because from this blastocyst, what we understand is that there are cells on the inside, which we call the inner cell mass, here depicted by these white circles. These are unspecialized cells that are fated to become the embryo itself, the developing organism. We can establish these cells in tissue culture. We can indeed disrupt this blastocyst and culture these cells in a petri dish, and they will grow forever. Now, some would contend that this is destroying the life, but the life of these cells persists in the petri dish. The embryonic stem cells are immortal. Now, we know from work in the mouse that these embryonic stem cells will undergo a continuous differentiation to form all of the cells of the body. And the embryonic stem cells are the most plastic cells of our stage of development. And that's to be contrasted with the more restricted fates of the adult cells. Once a cell is in an adult tissue, even though it's a stem cell, it is restricted to become the cells of that tissue and that tissue alone. Now, you may have heard that this principle of restriction, developmental restriction, has been called into question by certain papers recently published in Stem Cell Biology, papers that suggested that blood stem cells could actually become bone cells or muscle cells or fat cells or brain cells, and that brain stem cells could become blood. Well, this data was very provocative because it suggested that, in fact, we might not need to study embryonic stem cells if, in fact, we could simply coax this greater potency from adult stem cells. I want to say that over the last few years, much of this early data, many of these papers that suggested this plasticity of adult stem cells have been proven to be in error. In fact, if this plasticity exists, it exists as an artifact of cell culture, and it exists with such low frequency and efficiency that it's highly likely it would never be captured for any medical benefit. It has been called cellular alchemy. One has also heard that there are alternative, ethically acceptable alternative sources of stem cells, such as the stem cells that come from the umbilical cord. Well, we appreciate that the stem cells in the umbilical cord are primarily stem cells for the blood, and they can be used as a replacement for bone marrow in bone marrow transplantation. They, in fact, are yielding cures for leukemias and genetic diseases. But the stem cells of the cord blood are more like the restricted adult stem cells than they are like the pluripotential, unspecialized embryonic stem cells. Cord blood stem cells will never replace embryonic stem cells. It's an important principle. Why study stem cells? Well, what we hope is that stem cells will one day allow us to create novel medicines. But equally important is that through the study of stem cells, whether they're embryonic or adult, it allows scientists to decipher the rules by which primitive, unspecialized cells develop, diversify, and become specialized. This is enormously valuable as basic research. But we're already appreciating that this study of stem cells is starting to give us new insights into disease. So even without therapy, we're getting new insights into disease. And that's the concept of cancer stem cells. This idea that a single cell will self-renew and give rise to differentiated elements is typical of cancer. The cancer cell is a self-renewing cell. Now, there's essentially a model of cancer biology which suggests that the tumors are all derived from a single original cell called the stem cell. And it's that cell which self-renews and then differentiates. It's that cell which is the critical target of cancer chemotherapy. Unfortunately, most cancer chemotherapy simply kills cells non-selectively. It's a poison to all rapidly dividing cells. And what happens is the stem cells of the cancer tend to be resistant to the chemotherapy. So you kill the vast majority of cells. It appears as if your cancer is going into remission. But since you've left the stem cells behind, they will regrow and the cancer will relapse. So the goal of modern cancer chemotherapy, as this notion of cancer stem cells is evolving, is to define the specific molecular targets that distinguish the cancer stem cell from the massive differentiating, proliferating cells of the tumor, and find drugs that will kill the cancer stem cell uniquely so that ultimately the tumor will degenerate. It's through insights in stem cell biology that I think we're going to see major breakthroughs in cancer biology in the decades to come. This is human embryonic stem cells, colored with a green fluorescent protein that allows us to follow these cells and their progeny. These cells are grown typically on a background of mouse feeder cells. The mouse cells provide nutrients that sustain the growth and health of the human embryonic stem cells. Now, when we remove these green cells from their mouse supporters, they will aggregate. They will spontaneously form a ball of cells and start to differentiate, start to specialize into all the tissues that we recognize in the developing organism. Nerve cells, muscle cells, blood cells. Here, at day 14, these little islands of cells are developing red blood cells. Now, what scientists are doing is learning the principles that guide the development of these cells along specific tissue pathways so that with increasing precision, we can develop blood, as my laboratory is focused on, others focused on the development of pure populations of neurons. And imagine the complexity. Our brain contains probably no fewer than 200 distinct types of neurons. So the problem becomes one of figuring out the precise pathway along one of those neuronal subtypes. Other groups are interested in heart muscle and then the production of the insulin-producing beta cell. Then the hope would be, with stem cells in hand and differentiated tissues from them, that we might identify diseases that would be amenable to stem cell-based therapy. Well, the diseases that are most likely to be cured by this strategy have a few criteria, and I offer them to you. One is that they're likely to be caused by the loss of a single cell type. So we're not talking about entire organ failure. We're talking in particular about a disease where you're missing a particular cell within an organ or a particular tissue, and I'll talk about those. Another would be that we already have evidence that through either organ transplantation or cell transplantation that we can actually treat the disease. And finally, the unmet need would be that we would have a unique and inexhaustible cell source that would be provided by embryonic stem cells. What are such diseases? Well, one of the most important is diabetes, type 1 diabetes to be exact. This is an autoimmune destruction of the insulin-producing beta cells within the pancreas. It strikes children. It's a terrible disease. It means that that child is left with a lifetime of sticking their fingers every day to test their blood sugars, giving themselves multiple insulin injections, suffering the consequences of blindness, kidney failure, heart failure, and this is all due to the loss of beta cells. As I said, our colleague Doug Melton at Harvard recently had a very disturbing bit of data to prove that, in fact, the adult beta cell is not replenished from stem cells. This suggests that if we're going to have a source of beta cells for this type of therapy, we would need to have them from the embryonic stem cell. Now, we know that through whole pancreas transplantation, or in cases where you have a couple of cadavers and can isolate just the beta cells from those cadavers, that you can transplant those cells and make diabetics free of insulin for several years. So this is a loss of a single cell type. We know it can be cured or at least treated successfully with cell transplantation, and yet we don't have a good source of cells, and embryonic stem cells would be that source. Several groups worldwide are already publishing very exciting results with animal models and the production of beta cells from from embryonic stem cells. The second target, which gets an enormous amount of attention, is Parkinson's disease. Parkinson's is the loss of a single neuronal subtype in the midbrain, the neurons that produce dopamine. There's been 10 or 15 years of experience with transplantation of dopamine neurons taken from aborted fetuses, and those patients in many cases do sustain improvements in their motor dysfunction. Obviously, we would prefer an inexhaustible supply of dopaminergic neurons, and embryonic stem cells offers that. A group here locally led by Ole Isaacson has been able to successfully treat a rat model of Parkinson's disease with ES-derived dopaminergic neurons, and Ron Mackay's group at the National Institutes of Health have also recently reported very exciting data on human embryonic stem cell development into dopaminergic neurons. And then lastly, I would say that because we have had such success with bone marrow transplantation, and we can appreciate that the blood stem cell can be used to cure leukemias and genetic disease, we imagine that blood disorders could be treated with the products of embryonic stem cells. This is what my laboratory is particularly focused on. Now, it's not going to be easy. There are enormous challenges to treating patients with the products of embryonic stem cells. You may have heard that these embryonic stem cells, on the one hand, have this great immortal growth potential, which cuts both ways because that gives them the tendency to form tumors. And indeed, if you take the undifferentiated embryonic stem cells and inject them into an animal, it will form a tumor. It will form a rather benign encapsulated tumor called a teratoma, but it's a tumor nonetheless. If one were transplanting cells into the brain of a Parkinson's patient, a teratoma would be a devastating complication. So there are strategies that we imagine for adding suicide genes into cells so that once we transplant them into the patient, should a tumor occur, we can give a drug which would eliminate the cells. One strategy. The other strategy is that we be certain to coax the embryonic stem cells to form a specific differentiated cell type that would then be isolated and purified, free of the tumor-forming cells, and delivered to the patient. The second problem is that just as organ transplants face immune rejection coming from one individual into another, so would ES cell transplants. And overcoming tissue rejection currently requires poisoning a patient's immune system with very toxic immunosuppressive drugs. So there are strategies whereby we can combine the methodology of nuclear transfer, which I will describe, with embryonic stem cell biology to create autologous or self-tissues, tissues that would be identical to your own and would therefore not be rejected by your immune system. This is the process of nuclear transfer. It is a process of cloning, and I will take you through it and describe to you the distinct difference between the intention of this procedure for reproductive cloning, the cloning of a child, and the applications in medical or research cloning. Both procedures start with a donor cell. In this case, imagine yourself as a child with sickle cell anemia, and one would start with a skin cell from that child. You would have to have an egg, a donor egg. This is the egg derived often in the context of in vitro fertilization. And the DNA of that egg would be removed. And this is the process shown here in a video by Kevin Egan at Harvard. What you have is a holding pipette with gentle suction on the left and the egg squeezed against the holding pipette. You have this micro needle which inserts itself into the egg. Let's try that again. And what the needle is doing is withdrawing the DNA of the egg. What's left behind is simply the bag of cytoplasm within the egg, and it is within that cytoplasm that is contained a very fascinating group of molecules. The next step is to take the skin cell, the nucleus of the skin cell, and insert it into the egg. And it's simply the reverse of this process. What you'll see is the nucleus coming in from the side, being placed into the egg, and then withdrawn, and then the next egg is brought into place. Now, once the skin cell nucleus is within the milieu of the egg, a fascinating and remarkable process occurs. All of the features that make this cell a skin cell are erased. You appreciate that every cell in our body contains the same DNA. What makes a skin cell distinct from a blood cell is a set of genes that are unique to the skin cell. Well, in this process of nuclear transfer, the egg cytoplasm reprograms the genes that are on in the skin cell, erasing all the skin genes and reactivating the embryonic genes. This is probably the most fascinating process in biology today. It's something that we're only beginning to understand. But it allows us, in a sense, to trick the skin cell back into its embryonic state, and indeed it begins to divide and it does form an embryo. Now, this embryo can be transferred into a recipient animal, as was done with Daley. And as long as you have enough recipient animals and can do this, it took over 300 transfers to get one Daley, one can indeed perform reproductive cloning. And in fact, it is the worry of many in our community that this technology will be misappropriated to produce human clones. I want to say, and I'll speak more about this, that there is consensus among the scientific and medical community that this would be a crime. However, we do believe that there are legitimate medical applications of this technology which involve the extraction of the stem cells from within this early embryo and placing the stem cells into the petri dish, because with this process we create a customized stem cell which is identical in all means genetically to the donor of our skin cell. And if our intention is to treat a child with sickle cell anemia, we might start with this process, end up with an embryonic stem cell, make blood cells from this embryonic stem cell, correct the genetic defect, and repair that child's disease. What is so bad about reproductive cloning? Well, there's a wide moral debate about the propriety of copying us as individuals. But let's just talk about the science. We know from cloning 8 different mammalian species that cloned animals are abnormal. The vast majority of cloned animals die during gestation. Even those that are born often die in the immediate perinatal period, and those that are born and survive have a shortened lifespan and are often beset by significant medical problems. This is because this process of nuclear reprogramming is inefficient. It is not 100% complete. And in order to generate an entire organism with hundreds and hundreds of different cell types, any specific failure of reprogramming might lead to the failure of a specific cell type and lead to the death of the organism. So reproductive cloning is unsafe. That doesn't stop this from being touted by fringe elements within our community. And there are literal crazies who are advocating human cloning. There is a group led by His Holiness Rael. Here he is here. This is Rael. He is the well-known spiritual leader of the Raelian movement. This is the world's largest UFO-related organization. And they've founded CloneAid. This is the first company to offer cloning to human beings. Now, they were on the television last year claiming to have cloned a baby. I can assure you that no one is, first of all, scientifically ready to be able to achieve reproductive cloning with a human. And if they were to do it, I would suggest it would probably be impossible, as you would require literally dozens and dozens of women who would be willing to gestate these embryos. I just don't think it has been done. I don't think it will be done. And I think it's both morally and scientifically reprehensible. But the question is, what about tissues that are derived by nuclear transfer? Are tissues going to be normal? And we believe they are, and there are several scientific arguments for why it's easier to pattern a single tissue than it is to pattern a whole organism. And in order to test this principle, my colleague Rudolf Janisch and I decided to perform an experiment to ask whether we could marshal all of the technology of nuclear transfer to treat a disease model in the mouse. We started with a mouse that lacked an immune system. This is a mouse that is a model of bubble boy immune deficiency. And then using strategies similar to those that cloned Daley, tail tip cloning, we literally just clipped the tail, just the edge of the tail off this mouse, cultured some cells, and derived a embryonic stem cell line. Now, this embryonic stem cell line would be genetically identical to the mouse, carry the genetic lesion. And now once we have that line in the Petri dish, it becomes trivial to perform gene therapy. This is, in fact, one of the greatest advantages of using nuclear transfer because it makes gene therapy straightforward. Gene therapy has been the goal of medicine for the past 15 years. It was enormously touted as the next coming, the next solution for all kinds of genetic disease. We appreciate that the challenges are much more difficult than we had imagined. And now, 15 years after the first clinical trials, we're starting to see the treatment of kids, in fact, with very similar bubble boy type diseases. We're seeing success, but we're also starting to realize that willy-nilly introduction of genes into the genome carries the risk of activating cancer genes. And in fact, 2 of the kids in this first trial have come down with leukemia. Using this ES cell intermediate, we can perform gene correction with surgical precision. We can replace the defective copy of the gene with a correct normal copy. We're simply not adding a normal copy into the background of defects. We are correcting the gene lesion. And then using techniques developed in my lab, we can develop blood stem cells and transplant the mouse. This work was published 2 years ago. We published that it was partial correction of the immune defect, not a cure. But in the 2 years since we published this work, we've made significant advances in our ability to generate virtually complete immune reconstitution of the mouse. The technology is here today. It could be practiced. It's enormously cumbersome and time-consuming. It's not something which is going to be jumped upon by the pharmaceutical industry because no one's going to make money doing this. But as a proof of principle for the medical value of this technology, this experiment stands as a landmark. This experiment has given my group and other groups an interest in applying these strategies to treat a variety of malignant and genetic diseases of the bone marrow. Similar strategies could be used to treat genetic diseases that affect other organs. And indeed, we at the Harvard Stem Cell Institute have indeed set in motion a request to gain approval through the institutional review boards to actually perform this technique using human cells. Our ultimate goal is to treat disease and to relieve suffering, in particular from our strategy in kids with various genetic diseases of the bone marrow. This work has been successful with human cells. It's been successful once. The Korean group published last year that, in fact, they could marshal nuclear transfer and all of this technology to produce a cloned embryonic stem cell line. Just the beginning. I think we can go further. Now, I will spend some time talking about the ethical issues, and one of the questions I'm often asked is, why, if there are ethical issues surrounding embryonic stem cells, why do we need to study embryonic stem cells if there are alternatives, if there are stem cells from other tissues, in particular stem cells from the adult? Well, let me reiterate. As I said, there are indeed adult stem cells from the bone marrow, the skin, the gut. There are stem cells from the fat. They're very much like the stem cells that are common to the mesenchyme or connective tissue. But remember that these stem cells provide only a limited range of tissue types that can be generated. It's probable that there are no stem cells for the heart. If we want to repair failing hearts, the embryonic stem cell is the source. There are no stem cells for the insulin-producing cells of the pancreas. If we want to treat type 1 diabetics, the kids with diabetes, we need to use embryonic stem cells. The adult stem cells are difficult to grow and manufacture. And I would argue that for a variety of scientific reasons, that is the ability to answer fundamental questions of biology, just simply studying the stem cells in the Petri dish, adult stem cells will not replace the need for embryonic stem cells. But the science is moving very rapidly, and I think the ethical arguments are slow to catch up. The embryonic stem cells derive from human embryos. Indeed, they involve the destruction of those embryos. Now, most of the cell lines that are in use today are derived from embryos that are left over after couples have gone through in vitro fertilization. There are 400,000 of these embryos frozen in vitro fertilization clinics throughout the country. Now, most of those embryos, one hopes, will be used in the reproductive process and will give forth children. But many of the embryos will be discarded as medical waste, and many couples believe that they would prefer that those embryos be donated to medical research. Now, I think there's a misunderstanding among many in the public about what an embryo is. I've tried to describe to you that this is a microscopic ball of cells. In fact, there are no nerve cells. There's no body there. Some people vision a homunculus within these embryos. These are tiny microscopic structures, and it's the fact that they are so early in human development that makes them so valuable to scientists. But nonetheless, the issue remains, what is the moral status of these embryos? And the question is often asked, when does human life begin? Well, I should say that among major world religions, there is no consensus on this issue. Catholicism holds to the claim that life begins at conception. That means they assign moral significance to the conceptus, and they speak about the culture of life. Interestingly, in the Judaic and Islamic tradition, moral weight is not given to the earliest stages of human development. Moral weight is assigned only later in human development. The embryo, the human, is as like water until 40 days in the Talmudic tradition. And so major world religions do not agree on this. Now, Catholicism came to the notion that life begins at conception relatively recent in its history. It can be traced to a papal encyclical by Pope Pius IX in 1869. And it was clear at that time scientists were just beginning to visualize fertilization in under the microscope. And so there's no doubt that at conception a new genome is formed, a new compilation of DNA, a new unique arrangement of genes. But if we're going to use biology to inform when life begins, let me introduce some biology that causes one to question, to consider some of this fuzziness. Because even though at conception a new genome is formed, individuality of human beings is established later. Now, we appreciate that up to 14 days of human development, twinning can occur. The embryo can split as many as 8 times. Identical octuplets have been produced. And there is another equally fascinating concept, which is that 2 individual sperm can fertilize 2 eggs, 2 individual conceptions, and those embryos can fuse into 1 individual. There's a fascinating paper that appeared in the New England Journal in the last 2 years. A woman who developed kidney failure. And so they did tissue typing on her family members to find a match. And they went back to the woman and they said, we have some bad news. Your 2 sons, they're not your own. There's some disputed maternity here. Well, I mean, disputed paternity is a fairly common thing in tissue typing. But, you know, the mother was saying, well, geez, I remember the labor. I remember you could switch 1 child, but maybe not 2. Turned out she was a tetrachometic chimera. She was the embodiment of 2 conceptions. She had 4 DNA types. So it raises the question, if individuality is closely linked to spirituality, and individuality is not conferred until later in human development, I think it blurs the question of when personhood or ensoulment occurs during biological development. And I would argue, if I'm taking your cells and I'm simply changing the nature of your cells, reprogramming your skin cell back to its embryonic state, so that I can then coax those cells to become insulin-producing cells to treat your diabetes, when in the course of that transition of cells is a new independent life worthy of protection created? There's been arguments offered to the contrary that say that persons are members of a social community that shapes and values them, and personhood must be defined in terms of interactions and relationships with others. I put forth that life and personhood are 2 different concepts, and that we as a society must think about personhood, because fundamentally what this debate opposes are the rights of an embryo and the rights of individuals and patients whom this medical research and ultimate therapy might serve. President Bush has attempted to tackle this very challenging set of ethical issues. And he weighed in a remarkable speech now over 3 years ago, the first primetime speech of his presidency. And President Bush was very circumspect in deciding that he would in fact endorse embryonic stem cell research. Many in his conservative base wanted him to ban the research altogether. But I believe he appreciated that the value of the science was too great and had too much interest to too many in the society for him to suppress it. But he walked a political tightrope in that he allowed the science to go forward on only a very limited set of lines. And that has been, in fact, a very vexing problem for the scientific community. These lines are registered. They're listed on an NIH registry. There are 22 lines available. Now, President Bush had promised that there were 60-odd lines. Unfortunately, it turns out that many of the lines that were thought to exist, in fact, didn't exist. Many of the lines are owned by companies. They're companies that exist outside the United States. It's very challenging for United States scientists to get hold of these lines. And since August 9th, there's been an enormous amount of progress. Science doesn't stand still. This is an incredibly rapidly moving field. And to be constrained to using pre-2001 vintage cells is the equivalent of saying that you can't upgrade your computer system. There are over 128 new lines worldwide. Doug Melton has produced some of these lines. They're well characterized, they're easy to culture. Other lines have been grown free of animal contamination. There are lines that have been developed that carry mutations that represent models of human disease. These are enormously valuable for physicians and scientists interested in these diseases. And I would argue that the families of patients afflicted with these diseases have a very strong interest that we be able to study these. But of course, we cannot study them using our federal dollars. Now, President Bush put a restriction. He didn't outlaw it, but he said you can't use federal money. The vast majority of my support as a scientist comes through the federal government. The federal government is the most significant provider of funds for basic research in the world, and it's the reason why the United States enjoys such an outstanding scientific and healthcare system. So this work has been really completely bogged down in many ways in politics. You appreciate recently that California decided to buck the trends and have approved a $3 billion funding measure so that California scientists will be able to do this kind of research. It will make California the United States mecca for such research. The laws banning reproductive cloning in this country have been passed in the blue states, but there are even some states that have passed laws banning all types of research, including research cloning. So it's become a patchwork quilt of regulations. I think it'll only get worse. And this has been recently debated in the United Nations. And in fact, just today, the United Nations abandoned a treaty where there was consensus on outlawing reproductive cloning because they could not come to consensus on research cloning. So because we can't come to consensus about the medical applications of this work, we are not outlawing the practice of reproductive cloning, and I think this work will go forward. The goal here is to keep an eye on the patients that might be served. Daniel Perry, now at CAMR, a research lobby organization, has written that there are enormous populations here in the United States that will suffer from disease that can be served by stem cell-based therapies. But fundamentally, it comes down to a societal argument about the rights of the embryo and the rights of patients. And every one of us as individuals has an opinion, and I am eager to hear some of that opinion in the discussion period. It's clear that as we move forward with this science, the science of nuclear transfer is applied to medically relevant and important therapies, that it does lead one to question the slippery slope towards reproductive cloning. And this is, I think, the specter that scares many, many people. Well, I take a certain comfort in society's ability to do the right thing. I think that we as a society can decide that there are legitimate applications of science and that there are illegitimate applications of science, and that those applications be restricted and regulated. I treat cancer patients. I give my cancer patients the great relief of morphine for their suffering. Morphine is abused on the streets every day by drug addicts, and it is regulated. We do not outlaw the use of morphine for legitimate medical applications because of the risk of misappropriation. Now, my lab has been engaged in nuclear transfer studies, cloning studies in the mouse, and I have to say that I've also been engaged in reproduction the old-fashioned way. I have to be a little careful, my wife is here, but we have two beautiful kids. And I will say that reproduction the old-fashioned way remains infinitely more pleasurable than cloning, and I just simply don't think that we're going to have to face this specter anytime soon. So let me summarize. Embryonic stem cells are incredibly important research tools. They are already ushering forth important new information and fundamental knowledge about disease. And our hope is that one day they will usher in a critical new modality of cell replacement therapy, the era of regenerative medicine. Currently, however, we as scientists are operating under a political cloud, and we are losing precious time. I wrote a piece recently in the New England Journal of Medicine highlighting the missed opportunities under the current policy that is the federal policy as put forth by President Bush. I think I would encourage you to look on the New England Journal website and read about these missed opportunities. I am in no way trivializing the value of somatic or adult stem cells. My laboratory remains actively engaged in the studies of somatic stem cells, leukemias, and all that derive from the adult stem cells. But embryonic stem cells will never be replaced by somatic stem cells. And I would argue strongly, because of the value of creating customized tissues for patients, to be able to correct gene defects in these tissues, that research into somatic cell nuclear transfer and cellular reprogramming is indeed needed. If you want to learn more, there are websites which will answer your questions, and I look forward to taking some questions as well in the question and answer period. Thank you very much for your attention.
Thank you, Dr. Daley, for a wonderful presentation. Right now, what we'll do is we'll take until about 8 o'clock, taking general questions from the audience, and I'll ask Dr. Daley to please repeat that question into the microphone so everyone can be sure to hear it. Keep in mind, again, that this is being webcast on WGBH. If you do not wish to have your question aired, there will be plenty of time to talk to Dr. Daley and his research scientists afterwards. So if you will entertain some questions now. Thanks.
Thank you.
One of the theories about why Daley the sheep had problems is because her DNA was old due to inherited shortened telomeres. Why doesn't that happen when you create a new line of embryonic stem cells?
So the question was, might Daley have been sick primarily because she was cloned from an old cell, and cells contain chromosomes, and at the end of the chromosomes are these structures called telomeres. They're the tips. They're like the little ends of the shoelace that keeps your shoelace from fraying. These telomeres also keep your chromosomes from fraying. Well, the original publication suggested that Daley's telomeres were short. It was wrong. It was almost certainly wrong. Turns out that the cloning process, when you take an old cell and put it back into the nucleus of the, back into the egg, the telomeres actually are restored in most instances. It may be in part that there's a selection process as well. We know that it's a very rare cell that actually is successfully recloned. So part of at least one hypothesis is that we do 300 different nuclear injections, and it's really the rare cell whose telomeres are in pretty good shape to begin with that allows us to develop a cloned cell. But in general, the cloning process allows us to select for cells whose telomeres are relatively healthy.
My question was related, and that is, when stem cells renew, is there any degradation with successive generations?
So when stem cells renew, do they age, is really the question. And in fact, stem cells do age. So we know it anecdotally from my experience harvesting bone marrow. So we used to do a lot of bone marrow harvests in kids and adults. And you were always a little bit more enthusiastic when you were going after that bone marrow from the kid because it's juicier, it's healthier, it's more vital. You know, by the time you get to my age, you know, you've got a bit of decrepitness creeping in. And so if you have a choice, take the young cells, no doubt. Stem cells age. Stem cells probably are a bit of a hedge against the aging of many of our other tissues. Stem cells have a certain degree of telomerase activity more than differentiated cells. So stem cells do better than other cells, but they are still not immune to aging.
That would seem in and of itself to argue for new lines.
So the question is, one argument for the need for additional embryonic stem cell lines is that the embryonic stem cell lines that we have are destined to fail, destined to age, destined to become obsolete over time. I think, yes, that is one of the arguments. There are strategies that we typically use in the laboratory where we take a culture of cells, we expand them, and then we take a very small subset and we make sure it's healthy and we expand that. And by going through this repetitive subcloning process, we can usually keep the cells reasonably healthy. But there's no doubt that that's one good reason for needing additional lines, because for various reasons, the lines that we have can sometimes go off and become damaged. And so new lines are needed to sustain the research.
I don't know if this is practical at all, but as far as I can tell, the only way to end the ethical debate on using female eggs and sperm is to recreate them in a petri dish to grow an egg. So I'm just wondering if anybody knows exactly what an egg is, or if it's feasible to recreate them.
Can I get your name? And I want to. Do you need a summer job, son? This is a kid who's going somewhere. The question was, one strategy for answering some of the ethical challenges is to make eggs in the petri dish. And in fact, that's precisely one of the goals that my laboratory has. I think it's challenging if you imagine that if the only recipient cell for nuclear transfer is a human egg, that would require women going through ovulations, taking hormones, having egg retrievals. Women do this all the time for assisted reproduction, but to do it for research or for therapy is another question. So because I think that's impractical and challenging, we have asked, can we make eggs from embryonic stem cells? We tried very hard. As often happens in science, you get beaten. We were beaten by a team at University of Pennsylvania that made eggs. We actually went on and made sperm. We're now back in the lab working hard to try to make eggs. We think it's going to work. We do think that one day we will be able to develop a source of eggs from tissue culture cells, and that will take women, if you will, out of the equation, which is good, because it's not a I always have to be careful how I say that. I hope you appreciate my intention was good there. Well, look, when we published the paper that we could make sperm from petri dish cells, you know how many questions I got about, well, does that mean we don't need men anymore? Are you completely worthless now? And the reality is that actually you still need the Y chromosome to make a sperm. So we're not quite out to pasture yet. But no, I mean, think about it. I think that having a source of eggs could make this work practical. But really where we want to go, I don't think anybody, that process that I outlined of cloning is so cumbersome, so labor intensive, that it's not likely to be scalable to a large population. So what we need to do is we need to use the nuclear transfer process to teach us how to reprogram cells directly. I'd like to one day be able to take your skin cells, put them in the petri dish, and treat them with chemicals and hormones in such a way that they just immediately become an embryonic cell, or they immediately become an insulin-producing cell or something else. It's that science, which is the science of the future. That's where we want to get. And it's going to take us probably some time where we are using eggs, but I really hope one day that the scientific progress will solve the ethical debate, because we won't need to use embryos again. Over here.
First, thank you.
And great question.
Thank you for your work. All the things today. If one was interested in Parkinson's, a family member who has Parkinson's, where is the most promising research in the world? It's less of an academic research question for my family. I'm interested in what sort of treatment is possible.
Mm-hmm. Mm-hmm. Well, we are extremely fortunate here in the Boston area to be really one of the world centers for research and treatment of neurodegenerative disease in general, and Parkinson's in particular. We have one of the world's authorities on Parkinson's disease in Ole Isaacson, and in fact his laboratory is aggressively pursuing the generation of these dopamine neurons from embryonic stem cells. I think it's really exciting. There's a website for the Harvard Center for Neurodegeneration that you might want to consult. And Ole is also someone who speaks frequently in the community, and you might have an opportunity to hear him. It's, I think, extremely exciting work. Laurent Studer at Memorial Sloan Kettering has published a paper recently generating a very nice population of dopaminergic neurons from human embryonic stem cells. And I also mentioned Ron Mackay, and there are others throughout the world. In the back over here. Well, we're in an environment where at least the work can be performed and you won't be thrown in jail for doing it, which, I mean, really, 3 years ago when Bush made his announcement, that was one of the concerns that we all had, that there would be a prohibition that would restrict anyone from doing the science. However, the challenge now is actually getting enough funding to do it. And so that's part of the reason why we started the Harvard Stem Cell Institute, to put the stature and the power of a great institution like Harvard behind raising enough capital so that its scientists could perform this research. We formed the institute. There are over 250 scientists that have signed on as members. There are an initial round of grant applications which were submitted. There were 60 grant applications, and we only have enough money to fund 6 of them starting in January. So one of the real challenges is fundraising. And many of us are spending now a lot of time talking with donors, talking with individuals who have an enormous amount of interest in this area and also have the wherewithal to be able to pick up some of the slack. The problem is that I can't imagine philanthropy ever being able to make up the difference that would be needed to move this field forward as quickly as we'd like. But remember, this is an international effort. I was on a conference call today for the board of the International Society for Stem Cell Research. We're trying to decide whether we're going to have the 2006 meeting in Australia or Asia or Toronto. And we had scientists from Sweden and China and the UK and all. This work is exploding everywhere, and there are countries like Singapore and Israel and Japan and China that see this as the next frontier. They see it as a way of reaching competitive advantage over the United States and the West, and they see this as their mark, and they are going to invest in it in a big way. So the science will move forward. Unfortunately, it might not move forward as quickly here. David. That's our hope. That is really our hope. That's one of our goals. And it is a very, very lofty goal. Enormously challenging. But it's so exciting that I think lots of us are imagining ways of getting there. So, in fact, we're just beginning to ask the basic questions, comparing different populations of stem cells. So stem cells from the blood, stem cells from the brain, embryonic stem cells. And when you look with these very powerful techniques that are called microarrays, you can define the exact subset of the 25,000 human genes that are uniquely expressed in neural stem cells, blood stem cells, embryonic stem cells. It turns out that there's a very small set, maybe 500 to 1,000 genes that are common among them. Those are the signature of stemness, if you will. They are the genes that make a stem cell a stem cell. If we can understand how to activate those genes, which is what the egg is doing when it takes your skin cell and reprograms it, if we can understand the way the egg reactivates those genes, we might be able to do it in a Petri dish. And in fact, that's one of the goals that we have. I applied for an award through the National Institutes of Health to do that, and even though the National Institutes of Health won't be able to fund any of the human nuclear transfer work, they will fund this type of work.
Through your work with animal models, have you been able to isolate at what point in the pluripotent cell to a fully differentiated cell, the cell actually gains the ability to be rejected by the body to have an response? And if so, does that tell you anything in terms of specifically the ability of that cell to be able to go into other lines of cells?
That's a very interesting question. So he's asking, when you start with these unspecialized stem cells, and then you're making them into a specialized blood cell for transplantation or something, at some point along there, the immune system starts to recognize it. And at what point does that occur? It's fascinating. We're just starting to understand that problem. But the embryonic cells may in some way be immune privileged. I mean, if you think about it, when the embryo inserts into the mother's uterus, it is a unique individual, and yet the mother doesn't reject it. Trying to understand the tolerance of that maternal-fetal interface, it's a fascinating question. The genes, the proteins that are on the surface of all cells that are recognized by the immune system, actually aren't turned on until about halfway through our development in gestation. Now, unfortunately, our immune system not only recognizes foreign immune genes, but it recognizes the absence of immune genes. So if we just take our embryonic cells and they're not coaxed past the point where they're expressing the normal immune genes, even though they're genetically identical to you, they'll be rejected. And we actually saw that in some of our animal models. So you've asked a very, very important question. It's one that could require a very, very detailed and long answer, but it's a good one. We're studying it. If you need a job, come see me.
It might be a similar question. Can you explain the mechanics, just by mention often taking an embryonic stem cell and coaxing it into a very specific kind of differentiation? And as someone who is not a scientist and is amazed at the idea of these teeny little zygote ending up being us, I have no clue as to how it possibly happened. And what you have said is that there's actually some techniques by which you take that and guide it to not becoming a tumor cell, but actually becoming a blood cell.
Right.
The mechanics and the challenges of doing that.
Right. So how does one even imagine taking this blank slate, one of these unspecialized embryonic cells, and getting it to commit to all the specific intermediate points of differentiation to form an incredibly complicated subtype of nerve cell? If you think about it, it's a dazzlingly challenging problem. We take some comfort in the fact that the cells are much smarter than we are, as it turns out, and that these programs, in many instances, are preordained within the cell. And so what we do is we have to actually work to keep the cell in its pristine, undifferentiated, unspecialized state. We actually work hard to do that. If my people take the weekend off, the cells start to frolic and differentiate. So the differentiation is really, the default pathway. So what we do, we are just starting to learn how to not just step back, observe it, and then fish out the cell that we want. That's really what one can do. We're now starting to appreciate the genetic pathways that the embryo uses to specialize a certain cell. So we've been studying the genetic pathway and using model organisms like the fish. So we've defined genes that when they're turned on, we get a blood cell. And what we can then do is we can manipulate those genes in our embryonic cells. We can turn them on and off like a switch. And when we turn them on, we promote blood. When we turn them off, we stop blood, and we might even promote heart or something like that. So it's through understanding that immensely complex set of molecular switches that we can actually start this process. It's a science that's in its infancy. I'm amazed we can do as well as we can. I would say 10 years from now we'll do a lot better. I'm not supposed to answer that question. When are we going to see success? That's the hardest question to answer. It's the hardest question to answer. And we're always at risk by promoting the promise and the hope of over-promising and under-delivering. That's what happened to gene therapy. Gene therapy seemed so at hand in the late 80s. We could do it in animals. We couldn't even do it that well in animals, unfortunately. But people thought, the breakthroughs are just around the corner. And they promised, and they got patients involved in trials, and there was all this enthusiasm. And there was some overstepping of bounds by some investigators, and some patients were hurt. Some patients died. We're worried about replaying that in the stem cell field, and I think most scientists have tried to be very careful to say that the science is important right now for answering basic questions, and we should bring it forward, and that we can't guarantee when the clinical therapies will come. We just can't guarantee that. But if I had to have my arm twisted, I think there are going to be trials, there are going to be attempts to get into the clinic within 5 years. I don't predict those will be particularly successful. Because if you just look back at other major new technologies that have been introduced, take monoclonal antibodies. They were first published in 1975. It wasn't until the late 80s, early 90s that we actually had our first approved drugs that were monoclonal antibodies. And it's really only in the last 5 years that people really see monoclonal antibodies as viable viable drugs, viable medicines. 25 years. I think it's probably going to take us something like 25 years before people take stem cell therapies for granted. 25 years.
How would you like to see the laws change regarding stem cell research and funding? And what kind of regulations would you like to see?
What kind of regulations? First of all, I really think that given the nature of this work, there do need to be guidelines, there do need to be regulations. We just had a meeting in Washington. The National Academy of Sciences convened a panel, had scientists come and give presentations, and really ask what are reasonable and legitimate regulations. So there are issues of informed consent. Couples need to know if they're donating their embryos, if they're going to research. They need to know how they're being used. There need to be no financial incentives to donate those embryos or donate those materials. We need to know, there needs to be transparency in the process. Laws. Currently, there is a law, essentially, that outlaws the use of any federal funds for research on embryos. Now, that's, in general, what's preventing all kinds of federal monies being used to improve the yield of in vitro fertilization. It's really limiting our ability to ask many fundamental questions about early human development. So studies of early birth defects, studies of congenital diseases, many of these are limited because we can't study embryos. We can't study human embryos. And I think that that's a very narrow-minded vision for the government. Here we have a pluralistic society, and these are incredibly important issues. I mean, think about the research into birth defects, research into congenital disease, all of that. It's all being limited by these laws that prohibit the study of living human embryos. We have hundreds of thousands of these embryos generated daily. Our society has gone there. We had the debate 20 years ago about in vitro fertilization. We practice in vitro fertilization. We have to be able to understand that there's a difference between a cluster of cells in a petri dish and a child or an individual who is in need of a therapy. I think we as a society can come to an agreement, and our government can come to an agreement, that the best service of human health is to support the use of stem cells in the research.
At this point, I'll end the formal question and answer period. In just a moment, I'll have Dr. Daley introduce the members of his research team. Right now, from 8 to 9, please partake again in the refreshments in the cash bar. Please also note that restrooms are just down the hall, and we'll have about an hour for informal discussion with Dr. Daley and his research team. Let's thank Dr. Daley one more time, and then we will introduce the team. great. And if you will introduce.
Yes. Let me introduce them, and as I do introduce them, they should stand up. We'll start here with Claudia Langerke. Dr. Langerke is from Germany via Romania. She's a practicing clinical hematologist. She's come to my laboratory with a precise interest in stem cell research, and she's particularly interested in cancer stem cells. Dr. Kiwi Liu, Taiwanese, educated at Rutgers in New Jersey. She's doing some very, very exciting work on the using embryonic stem cells to create cancer models. Willie Lench grew up on a pig farm in Utah. Educated at Oregon State, and Willie is studying human embryonic stem cells. He's doing some incredibly exciting new work in creating new disease models with human embryonic stem cells. Holm Zars, also from Germany. Holm is the ultimate guru in our laboratory with genetic modification of embryonic stem cells. And Azam Mohammed from India, recently here of the United States. And Azam is a world expert on a particular drug that is used to treat a leukemia, which is the classic malignancy of the adult blood hematopoietic stem cell. And Azam has taught us enormous amounts about the mechanisms of resistance to this drug. They're a brilliant crew. I'm proud to have them, and I'm eager to have all of you meet them and ask individual questions. Thank you.